Okay, so hopefully you've seen the grades are posted. So midterm one, the average was 84. You can pick up the hard copies of exams and should in the teaching lab. So there were 4 questions that were tossed out. So class policy is this: if 50% or more of the class gets a question wrong, then obviously there's a problem with the question or how it was taught. So I go back and add back those points. So as you look through your exams, check my arithmetic. Make sure that I added back points. If I didn't or made any kind of a math error, let me know and I'll be happy to correct it. So pick those exams up anytime. That lab is locked right now, actually. So if anyone wants to pick them up today, let me know and I'll get them for you. And then just a reminder that the next exam is actually coming up remarkably fast. So the next exam is not over as much material, as many slides or as many lectures, but the material is a bit more complex. So we'll make the second exam over a little bit less material. But that's going to go through lectures 14 through 21, which started on September 23rd and will end next Wednesday. That will basically be two slide sets. Okay, so we left off, was there a question? I'm sorry. We left off talking about the SOS response in bacteria. And so I mentioned that this repressor protein, LEX-A, regulates the entire suite of genes that are part of this regulon. It's probably about 21 or so genes and operons spread throughout the E. coli chromosome. That in the case when single-stranded DNA is present as a signal of DNA damage, my favorite enzyme REC-A becomes activated and it turns on LEX-A. In other words, it becomes a coproteolytic enzyme that degrades it, which releases these other genes, part of the SOS regulon, from repression. But it doesn't do them all at the same time. So some genes are turned on first. So genes like the UBR ABC system, the nucleotide excision program, get turned on first. Other gene products are turned on slightly later. Sol-A, for instance, is a protein that's important in cell division. So what the bacterium does by turning on Sol-A is it stops cell division. It's a cell cycle check type of a protein. It gives the bacterium time to repair the DNA before it actually divides. And then if the situation is very dire, if the earlier forms of nucleotide excision repair, for instance, don't take care of that single-stranded DNA's presence, then the bacterium will bring out the big guns, which are things like this UMUDC. This is an error-prone DNA polymerase. We'll talk about this a little bit, this is a type of DNA polymerase that really can make new DNA without actually having a template that it can read. So obviously, if you don't have a template and you're just making stuff up, chances are you're putting in wrong nucleotides. So this is the big gun that only wants to come out towards the end. So just another cartoon way of looking at this. Here's a normal situation shown on the top. You have, here's a replication fork. The bacterium is replicating its chromosome here. And elsewhere around the chromosome, you have the LEX-A gene here with its own operator. The LEX-A protein is shown by these blue hexagons. And it's binding to operator sequences. These O's stand for operator sequences. These genes, I don't like this part of the cartoon because I don't like reading from right to left, but this would indicate that the gene REC-A, for instance, is transcribed from your right to left. So the operator would be tending to stop, or LEX-A bound to that operator site would be a repressor. It would be tending to hold back the transcription. But it's leaky. It doesn't, it doesn't stop transcription completely. So some REC-A is going to be expressed, the same with UBR, A, B, C, and some of the other members. But that situation changes rapidly when DNA damage occurs, as shown in the bottom here. So ultraviolet radiation has hit this E. coli chromosome and has caused this famous thymine-thymine dimer. So that now what happens is that DNA polymerase could not move past that. It left a single-stranded region of DNA available before it could start resynthesizing the replicating chromosome. So that single-stranded DNA is an alarm. That's not a normal situation. So REC-A binds to that single-stranded DNA, becomes an activated REC-A protein, which now has the ability to cleave LEX-A. It's actually really, I'm oversimplifying this coprotease activity of REC-A. I'm oversimplifying it, quite frankly, because I don't understand it as well as I should. Because REC-A, while it is activated, and it does take on enzymatic activities, it actually, in the activated form, when it binds to LEX-A, actually causes LEX-A to cleave itself. So there is an enzymatic activity associated with REC-A, but it somehow turns on an autoproteolytic activity in LEX-A. So it's a very complicated system. But the end result is that LEX-A is destroyed, and then over time, you'll begin to express more REC-A, more UVRA, more UVRB. This is the SOS response. Save our ship, the chromosome has been damaged. Now, I mentioned that there are some heavy hitters, some DNA repair mechanisms that are held in abeyance because while they might save the life of that damaged bacterium, they might also cause such tremendous DNA damage that the surviving cell may in fact be toast. It may simply not truly survive. So UNUDC was nicknamed by a graduate student who discovered this, and the nickname has stuck. It's still referred to oftentimes. Even in the serious scientific literature, you'll sometimes hear it referred to as the sloppier copier. It's actually DNA polymerase 5, so E. coli has 5 different polymerases. Numbers 2 or 3, I can't remember which one does the majority of the replication in E. coli. But polymerase 4 and polymerase 5, DIN-B, are also referred to as DNA mutases. So these are part of the SOS response, but they're a late part of the DNA response. If the early responders, the first responders can't take care of the damage, then these guys come out. And they're going to fix damage if they possibly can, but they're going to put in a ton of mutations along with it. So there's an advantage to having this induced system to generate mutations, because while we're talking about the SOS response in response to DNA damage, because that's what was first discovered, it turns out that these systems are actually under the control of things that don't even damage the DNA. For some reason, you can treat E. coli with ampicillin, penicillin, that beta-lactam antibiotic, which interferes with peptidoglycan synthesis. And for reasons that no one still to this day understands, that will actually start the SOS response to some degree in E. coli. There are nutritional stress. If you put E. coli or many other bacteria under very starving conditions for a long period of time, for reasons, again, not completely understood, that will induce the SOS response. So while we talk about it for the most part as a DNA repair mechanism, not all of the things that bring the SOS response to bear actually have anything to do with DNA damage at all. So nutritional stress, is believed to trigger the SOS response as kind of this really crazy last-ditch attempt to try and utilize an energy source that maybe the bacterium couldn't utilize before. So just because a bacterium is in an environment where it doesn't have anything to eat, doesn't mean that there isn't some carbon or energy source out there that it just doesn't have the capability to utilize. So the thought is that nutritional stress induces these DNA mutases with the hopes that the bacterium will mutate some type of an enzymatic pathway that might allow it to utilize a new carbon or energy source. I mean, that's like winning the lottery, right? The chances of that are infinitesimally small, but bacterial numbers are huge. So it's possible that somebody in the population can now use a previously unusable energy source. These mutant cells are very infrequently viable. So for the most part, these mutants have mutations scattered throughout the chromosome. So while you might get an advantageous mutation at one locus, chances are you've got lethal mutations at 4 or 5 other loci throughout the chromosome. So the bacterium is probably going to die. So this hypermutation state that occurs in cells under stressful conditions is actually deleterious for the individual cell. So it's disadvantageous for that individual cell. Now, there's a couple of slides that I put in. Really, I'm updating for next year, so these slides aren't part of your PowerPoint. But let's go through them because I want to talk a little bit about UMUDC, this sloppier copier. So just as we said before, Rec-A, when it finds single-stranded DNA, can now help Lex-A cleave itself. And so if that goes on long enough, this UMUDC, the sloppier copier operon, so 2 genes are transcribed, UMUC and UMUD. What this system does is a type of DNA replication called translesion synthesis, shown on the top. This system, UMUDC, allows for the synthesis of a new strand of DNA when normal DNA polymerase can't get past this thymine-thymine dimer. And it's not just thymine-thymine dimers. I wish somebody sometime would put in some other lesion, but I'm tired of thymine dimers. I mean, you can have depurinations, depyrimidate, you can have other chemical modifications of the DNA that stop replication. It's not just thymine dimer. But in this particular case, UMUDC can now get past that thymine dimer, but you'll notice that it just puts in any random nucleotide that it feels like at that particular moment, just to keep things going, just to get the cell alive. So these are clearly not A's, as should be put in there. So this is going to be a mutation. And if these mutations have occurred throughout the chromosome as a result of the DNA damage, then you can imagine that these mutations are all over the place. And if the damage here was not just to these 2 thymines, but the bases beyond them were also damaged by a chemical. So aflatoxins, fungal toxins, are famous for damaging DNA. Reactive oxygen species, things like hydrogen peroxide, reactive singlet oxygen, hydroxyl radicals. These are all things that damage DNA. So you can have a stretch of DNA that was completely unreadable by a typical DNA polymerase. Yet UMUDC will just put in random base pairs, just doesn't care, puts in whatever it wants to, but hopefully the cell survives. But probably at a serious disadvantage. The UMUDC system is really actually pretty cool because there's Rec-A actually has a proteolytic activity specific, not just for Lex-A, but for the UMUDC system, specifically UMUD. So what's shown in this picture from a classic paper from 1998 is that when you have Rec-A damaging Lex-A, so that the repressor is removed, you get transcription of the UMUDC gene pair, that operon. UMUD, the protein, once it's synthesized, can actually be proteolytically cleaved by activated Rec-A. That's what the asterisk stands for. This is Rec-A that has been modified in its activity by having bound to single-stranded DNA, creating a slightly truncated version of UMU, shown here as UMUD prime. Now, UMUD prime or UMUD, the unmodified form, can interact with UMUC to form different versions. You can have one where you have 2 intact versions of UMUD, shown here on the left and the bottom, and 1 UMUC. That's actually involved in cell cycle regulation. This is another means whereby the bacterium will pause cell division at this particular point to try and allow the bacterium to repair its DNA. No one really knows what the UMUD prime C, so where you have both the modified and unmodified form of UMUD, no one really knows what that one does. But this is the one that actually does the mutagenesis. So the shortened versions, the Rec-A modified versions of UMUD, the D prime, interacting with UMUC, This is the translesion machinery. This is literally the sloppier copier. But depending on how these things get put together, it's a very efficient way to stop cell cycling and to do DNA repair with the same machinery. So UMUDC is a really fascinating means of DNA repair and survival. Now, we've talked about this SOS response in response to some kind of a stress. DNA damage, nutritional stress, antibiotics. But at some frequency, within any normal healthy population of bacterium, some cell just loses it. Some cell just turns on the SOS response for kicks. I don't know, just randomly turns it on. And generates what's known as a mutator cell. These are very rare. It's a suicide, really. I mean, this is the cell just giving up and generating lots of mutations throughout its chromosome. The cell itself is not going to be viable anymore. But so while that cell might die and degrade and release its DNA into the environment, that's actually believed to be the point. This is a process that's very controversial. Evolutionary biologists hate this term, but it's a term that microbiologists use called altruistic lysis, meaning lysis for the good of the population. So this fragmentation and DNA release actually end up being a way to help out the population. Evolutionary biologists hate this because they always want to say that this is called group selection. And group selection is something that just, it really doesn't occur in biology. But this is actually kin selection. Because think of that population of bacteria where one cell became a mutator, generated lots of mutations, died, and has now spilled its DNA into the environment. All of the cells around it are likely are genetic clones. So in essence, family members, identical twins, if you will. Now, into that environment has been spilled all kinds of DNA from that mutator cell that has mutations in it, most of which are lethal, most of which are deleterious, but maybe one or two are actually advantageous. So this is what altruistic lysis is all about. Because while we haven't covered this point yet, we will cover this in excruciating detail in another slide set. But bacteria evolve, and archaea as well, evolve remarkably differently than eukaryotic cells. You all know this to some degree. Bacteria are very famous for being able to take up exogenous DNA from the environment. In some lab, I guarantee you at some point in your biological science history, you've done transformation. You've taken plasmid DNA, most likely, and added it to E. coli and seen a change in E. coli's phenotype as it takes up that DNA from the environment. Transformation. Well, bacteria do this wildly in the environment. So the idea is that these cells that have lost control of the SOS response, mutated their DNA, released it into the environment, that DNA gets shredded. It gets broken down into pieces. So that while a bacteria might pick up an advantageous fragment, the chances are if that DNA has been fragmented, it doesn't also contain an allele that's lethal. So the lethal alleles that kill the bacterium now are hopefully separated just by random chance. There's hopefully some piece of genetic material that has an advantageous mutation that makes it into some member of that population and gives that member of the population an advantage that it didn't have before. This is part of the reason why bacteria can evolve so rapidly. This is the way that they generate, or one of the ways that they generate genetic diversity in bacteria is to use these systems. So these mutases are really important in terms of the evolutionary biology of bacteria. If you look at a graph of population fitness on this axis and mutation rate on the x-axis below, you really want to have kind of a sweet spot in terms of the mutases, the UMUDC, the DINB, being turned on. If they're turned on too little, too infrequently, then you have no evolution. You have no adaptability. If they're turned on too frequently and left on for too long, then you have tremendous genetic breakdown. so the mutation rate would be too high. So it's kind of like Goldilocks, the porridge is too hot, the porridge is too cold. You want to find that midpoint. So that's what the SOS response is really all about, is trying to find that sweet spot where mutases are expressed at just the right frequency and the right number of cells of a population to maintain evolvability without actually killing the entire population by putting too many mutations in. Okay, so that completes that slide set. So now let's move on and talk about my favorite topic, which are biochemical regulatory mechanisms. So we've talked a little bit already about transcription. And so now really what we're going to talk about is the ability of bacteria to regulate their transcription, because they can do it a little bit differently than prokaryotic cells. Than eukaryotic cells. So we've already touched on this point, but bacteria and archaea can have their genetic material arranged polycistonically. So more than one gene can be transcribed onto a piece of messenger RNA at the same time. So we're going to talk in great detail in this section about the tryptophan biosynthetic operon. But I'm just going to throw it up here as an example right now. So here you have 5 different genes that encode 5 different proteins, each of which is necessary to synthesize some member of the pathway to synthesize the amino acid tryptophan. They are all transcribed from one promoter. So RNA polymerase is binding here. It transcribes to the right, and you get one piece of messenger RNA. Now, that piece of messenger RNA can and is translated into 5 different proteins. This is something that eukaryotic messenger RNA never does, and we'll see why. In front of each one of the genes in a bacterial operon, there has to be an individual Shine-Dalgarno sequence. So if I ask you a question on an exam where I give you a 16-gene operon, and each one of those genes encodes a protein, and I ask you, how many Shine-Dalgarno sequences will you find in there? The answer is 16. There is one ribosome binding site for every one of these protein-coding genes. So there's a start codon to make at the messenger RNA level to put in the first methionine. But before that, there has to be a place where the ribosome binds. That ribosome is going to fall off at the stop codon at the end of this first gene, TRPE, making the protein TRPE. But another ribosome has to bind on to another Shine-Dalgarno sequence for TRPD, another one for TRPC, B, and A. This doesn't happen in eukaryotic messenger RNA. In eukaryotic messenger RNA, a ribosome binds to the extreme 5' end, translates until it hits the appropriate stop, or until it hits a stop codon and dissociates. There's no way for a ribosome to bind internally in eukaryotic messenger RNA, or I should say almost never. Some viruses have found a ways to trick eukaryotic ribosomes to bind internally, but that's just a virus being tricky. So these will have individual ribosomes bind in front of each one of the genes. Now, remember that this promoter is actually, and you can see it in this cartoon, the promoter region is not part of the transcriptome. So the promoter is where the RNA polymerase binds, but you'll notice that that messenger RNA does not include the promoter. So it's a common mistake that people make to think that the promoters are transcribed. Now, the reason that, or perhaps one of the reasons why operons are so efficient and why bacteria makes such wide use of them is that you can, by having 5 genes all creating the biosynthetic pathway for tryptophan, you can regulate them all simultaneously. You don't have to regulate TRPE on one part of the chromosome and TRPD on another part. If you need tryptophan, you can increase the transcription of all of the genes necessary at one time. If tryptophan is at very high levels, you can stop the transcription of all of the genes with one event. So this is a very efficient way to do transcription. To do regulation of transcription. Yeah, and I already mentioned that each of the genes has its own Shine-Dalgarno sequence. Sometimes that sequence is actually in the previous gene. The genes are so closely packed in the chromosomes of bacteria that in some cases the genes actually even overlap slightly by just 3 or 4 or 5 base pairs. Never much more than that. One of the things that bacterial chromosomes are famous for is their economy. Bacteria, archaea, and viruses waste very little space. Most of the DNA is coding for protein genes or regulatory or structural RNA genes. There's very little non-coding sequence in prokaryotic genomes or in viral genomes for that matter. That's very different than in eukaryotes, especially multicellular. Now, a word about the term operon. So the term operon is very widely used. It's a little bit archaic. Operon, the term was initially used by Jacob and Monod, the two famous guys who won the Nobel Prize in 1965 for their work on the lac operon. So they termed this assemblage of genes, the lac operon, as an operon because it had a sequence that they called an operator. Not all operons have, as it turns out, operators. The lac operon does, and many do, but not all of them. What operator sites are, and we're going to talk about the lac operon ad nauseum, but the operator sites are sites where repressor proteins bind. So we talked about the operator site for LEX-A in the SOS regulon. Those were operator sites. In front of each one of those genes in the regulon of the SOS, there was an operator site that bound LEX-A that repressed the expression of those genes. The same thing here. This operator sequence in front of the genes for the structural proteins of the lactose operon is bound by a protein that's not part of the operon, made by this LAC-I protein, or LAC-I gene here, which is independently transcribed into messenger RNA, then into a protein which can multimerize, bind to the LAC operator to help stop the transcription of the LAC operon. But my point at this point in our conversation is that this is where the term operon comes from, but now we know a lot of operons that don't actually have operators. So it's a little bit of an archaic term. So let's compare messenger RNA between prokaryotes and eukaryotes. So it's very different. So here is a messenger RNA from a bacterium that's polycistronic, gene 1 and gene 2. So there's a Shine-Dalgarno sequence here for the ribosome to bind and another one for here to bind. The way that prokaryotic ribosomes find the Shine-Dalgarno sequence is that they bind their ribosomal RNA to the messenger RNA at that Shine-Dalgarno sequence. Remember from your basic biology class, the structure of a ribosome is not simply a collection of proteins. It's a collection of proteins that are scaffolded upon structural RNA. 16S RNA, 5S RNA. You remember those, and then it's the same thing in eukaryotes. There's different lengths, different Svedberg units, but they also have RNA as a structural and an enzymatic component of ribosomes. So ribosomes are a ribonucleic acid complex. In bacteria and in archaea, the way that a ribosome is directed to bind to the right sequence is that there's a complementary sequence between the bacterial ribosomal RNA and this Shine-Dalgarno sequence. So there's actually base pairing between those two RNA species. That's what initially locks the ribosome on at the appropriate spot, just upstream, usually about 6 to 8 nucleotides upstream of the start codon. So ribosome will bind to the Shine-Dalgarno sequence by RNA-RNA interaction, which is weak, right? It's just hydrogen bonding. It can then start to translate at this start codon, usually AUG, but remember it can also be GUG or UUG infrequently. It will translate through until it hits one of the nonsense codons or stop codons. Those terms are used interchangeably, UAA, UAG, and UGA. amber, opal, and umber codons that will cause the ribosome to dissociate and fall apart. Translation stops. So if the second gene is going to be translated, it has to have another ribosome, and it's usually a different ribosome, binds to its Shine-Dalgarno sequence, again by RNA-RNA interaction, and it translates gene 2, and then it falls off. And this can happen 17, 30 times, depending on how many genes are present in that operon. Contrast this with what goes on with a eukaryotic messenger RNA. In eukaryotic cells, messenger RNA always has this very specialized 5' nucleotide cap called a 7-methylguanosine cap. This is what it looks like. You'll never need to draw it, just showing it to you for the chemists in the audience. But this 7-methylguanosine cap can only exist on the 5' end of messenger RNA. It can't exist internally, because if this were put internally, you wouldn't be able to add more nucleotides onto that. So this is always at the 5' end. This structure is what a eukaryotic ribosome binds and loads to. It has nothing to do, so far as I know, with an RNA-RNA interaction between the ribosome and the messenger RNA. In eukaryotic cells, the ribosome is recognizing this. It loads, it translates until it hits a stop codon and it falls off. And if there happened to be another gene downstream, there would be no way for a ribosome to get a hold of it, because the only way a ribosome can load is with this 7-methylguanosine cap present in front of it. So very different ways that eukaryotes and prokaryotes translate their messenger RNA. Now, just briefly want to go through, this is just a topic that microbiologists like to argue about while we're drinking at meetings. Why is it that, operons have evolved? And so there's 3 basic theories. So we've already touched on one of them, the theory of co-regulation. If you have all of the genes necessary for a pathway all assembled in such that they're all being co-transcribed at the same time, you can regulate that very well with a single regulatory event, rather than spreading the regulatory events out around the chromosome. So the theory of co-regulation. There's a theory called the subunit diffusion theory, which goes back to when the Earth was a lot warmer than it is now. So in a highly thermal environment, things in an aqueous environment diffuse more rapidly. So they tend to get away from one another or approach one another more rapidly. So if you were trying to create proteins from messenger RNA, if they were distinct messenger RNAs, yet those proteins needed to interact as a multi-subunit enzyme, like think of RNA polymerase or DNA polymerase, something where multiple proteins need to assemble in order to accomplish their role. If that's occurring in a highly thermal environment, then maybe it's more difficult for those proteins to associate. It's kind of a weak hypothesis. We don't like that one very much. The more interesting one is this theory of selfish genes. So we all know that DNA is a selfish molecule. So genes of a particular function tend to oftentimes in bacterial operons be clustered. This is not a hard and fast rule. You can find operons with a lot of different genes, and some of those genes, the proteins that they encode for, have nothing to do with the proteins encoded at the other end. But oftentimes what you'll find is that 2 or 3 of the genes encoded in there are actually of a related function. So here's another example. Here's an operon that's found in E. coli that allows E. coli to utilize the sugar galactose. We won't go through the pathway, but basically bacteria, in order to use galactose, to change galactose into a form of sugar that can feed into the glycolytic pathway, it has to have 3 different enzymes, the polymerase, transferase, and galactokinase. Now, remember that I said that bacteria tend to acquire DNA from their environment quite readily. If you were a bacterium that went through, that just died and spilled your guts into the environment, but you had this unique ability to utilize galactose, if your genes for these 3 enzymes were scattered throughout the chromosome, chances are no single bacterium would pick up all 3 of those pieces of DNA. So from those DNA genes point of view, they're lost, they're dead, right? Because if a bacterium picks up the galactokinase gene by itself, that does nothing for the bacterium that received that gene. However, from the gene's selfish point of view, if they by random chance over the course of their evolution have rearranged themselves into the chromosomes such that they're now adjacent to one another, they're now at a higher frequency going to be all contained on the same piece of DNA after that cell dies and releases its DNA into the environment. So now, if this entire piece of DNA gets into another bacterium, it gives that bacterium the ability to use galactose. But from the DNA's point of view, that bacterium is now more fit. It's going to divide more frequently. It's going to survive for a longer period of time, which from the gene's point of view is a good thing. So this is DNA acting greedily, acting selfishly. So this is probably one of the main reasons why bacteria, because bacteria take up DNA from the environment, why they've evolved to put many of the genes in the same functional category together into operons. It makes the genes more fit. They can be more easily passed on to other bacteria. Okay, now let's talk about what I promised that we would talk about in this set, and that's biochemical regulatory mechanisms. And I'm going to have to apologize right up front because I hate this terminology. There is some very archaic terminology that is so embedded in the literature that we can't get rid of it. And two of the terms we're going to deal with now. One is enzyme repression. Enzyme repression sounds like an enzyme, a protein, is being repressed. And it's not true. That's not what enzyme repression in this context means. Here, when we're using the term enzyme repression, it's transcriptional control. It's the control of how frequently transcription is initiated. Those transcripts oftentimes encode enzymes. That's where the term enzyme repression came from. This terminology goes back to it probably predates Jacob and Monod in the 60s, back when people didn't really understand completely how enzymes were repressed. But this type of repression, enzyme repression, is a very effective means of controlling transcription, particularly for biosynthetic genes that are involved in pathways that create things. So here in this hypothetical example in this graph shown on the right, we're looking at genes involved in the synthesis of the amino acid arginine. So we're looking at the relative increase on the y-axis over time on the x-axis. And so you can see that over time, cell number is increasing in a linear fashion. So this is logarithmic growth. The total amount of protein in that culture is increasing at the same rate. And so are the biosynthetic enzymes that make up arginine until a point. At this point where this arrow is, an investigator has added a slug of the amino acid arginine. Now what happens shortly after that is you see that the bacterium stops making the biosynthetic enzyme, so it goes to flatline. This is perfectly logical. This makes perfect sense. Why bother to transcribe and then translate the genetic material to synthesize arginine when you're swimming in it? You know, just import the arginine and save your energy rather than create that. So this kind of repression of the transcription of these genes is called enzyme repression. It's not really all that rapid in terms of the response, because at the point where that slug of arginine was added, there was already a ton of transcripts for those genes. There was already a ton of the enzymes translating from those transcripts, making arginine, and those have a life, right? They have a half-life, probably is a more specific way to say it. They have to be turned over. They have to be broken down over time. So there's going to be a period of time that even when arginine has now been added to the environment, the cell is still probably creating some of its own arginine. But it stops transcribing the genes for the arginine. So until those transcripts break down, it's still going to translate them. And until those proteins break down, it's still going to make arginine. So we're going to talk in a little while about feedback inhibition, which is actually probably what you would think of as enzyme repression. In feedback inhibition, you'll see that the preformed enzymes can actually be inhibited by the end product. So perhaps the enzymes that synthesize arginine can sense arginine presence and stop their enzymatic activity. That's very rapid. And that will occur even in this same pathway. But for the moment, we want to talk about the mechanisms of transcriptional control that mediate arginine biosynthesis. So we're going to stay with the model of arginine to explain enzyme repression. This is a means of negative control. So kind of like the lac operon, the lex operators of the SOS response, arginine biosynthesis is controlled by an operator sequence that's bound by a repressor. So what's being shown here is, here's the arginine promoter. So this has a -10 and a -35 site. RNA polymerase with a sigma factor will recognize the -10 and -35. They're not labeled in this. But RNA polymerase will now proceed to the right to transcribe our G, B, and H. Those three genes encode three proteins that are necessary to act one with another to create arginine. Now, this is all well and good if arginine is at low levels. If the cell needs arginine, this is exactly what it wants to happen, what's shown in A. It binds, it transcribes, the transcript gets translated into the proteins, arginine levels rise over time. That's a good thing. But when arginine levels reach a point where it's arginine sufficient, arginine replete conditions, it doesn't need to produce arginine anymore. It would be a waste of resources to do that. So enzyme repression kicks in, and now arginine actually binds to a repressor protein. So here in A, you see the repressor protein, but it's in a conformation. The protein is in a conformation such that it cannot bind to the operator sequence. This DNA binding protein, this repressor, as it exists in an APO form, APO repressor form, meaning it's not bound to arginine, is in a conformation that prohibits it from binding to the operator. So there's nothing stopping RNA polymerase. But as arginine levels rise over time, arginine now binds, and this is not a covalent modification, not a covalent binding. As arginine increases in concentration, the pool of arginine repressor shifts from the APO form of the repressor that doesn't have arginine, to that which is bound by the arginine, which at this point, a general term for this would be that arginine is a co-repressor. Arginine binds specifically to this repressor protein, allosterically modifies it, that is, it changes the conformation of that protein, such that in this now active state, it can bind to the operator, and that operator, the location of the operator is key. Operator sequences are always just downstream of the promoter, or many times they are right on top of the promoter. But in either case, when you stick this DNA binding protein onto that operator, it physically stops, it prevents transcription by RNA polymerase. So arginine transcription levels will drop, the protein levels will drop, and arginine levels will drop. And then eventually the system shifts back to what you see in A. So this is enzyme repression. But what's really being repressed, if you want to think of it, so if you want to think about how to make enzyme repression make sense, it's repressing the RNA polymerase enzyme's ability to transcribe the arginine biosynthetic pathway, the operon. Now, let's contrast that with another term that I hate that's also from the historic literature because it sounds, again, like something is acting on a preformed enzyme. Enzyme induction. Hate this term. But because it's like enzyme repression. Here, what we're really talking about, although it doesn't sound like it, is the induction of transcription, the increased transcript of messenger RNA. So this is also a very good means of transcriptional control inside of bacteria. It increases the amount of messenger RNA for the genes in a pathway. This is the best example of this, and we'll spend a lot of time talking about this, is the lactose operon. So enzyme induction is usually used to control genes in a catabolic pathway. That is a pathway that breaks down an energy source. It makes sense. You don't want to produce the enzymes necessary to break down lactose if lactose is not there. So you want to have a system where the genes that encode that breakdown pathway are controlled by lactose. So again, here in this graph, you see the relative increase in either cell number or total protein is occurring linearly as this cell grows over time, or this population grows over time. And if you look at the genes, the transcripts, or even the proteins for the lactose operon, they're basically almost non-existent, almost non-measurable over time, until some investigator adds a slug of lactose at the point indicated by this arrow. Shortly after that time, you can see this rise in the enzyme beta-galactosidase. That's the enzyme that breaks down lactose into glucose and galactose, the two component sugars. But it doesn't make sense to transcribe those genes until lactose is present. So this enzyme induction can actually use repressors, just like the arginine repressor does, or in other cases it will use activators. So we're first going to talk, what are we first going to talk about? We're first going to talk about the lac operon. So we're going to come back and forth to the lac operon several times. But this is an example of, and it gets confusing, I know, enzyme induction being mediated by a repressor. So induction's a positive thing, right? So something is induced, it's now synthesized at a higher level. And repression is the opposite connotation, right? It's like when something is repressed, it's not made as much. So in some ways, the lac operon, although it's enzyme induction, it's a little counterintuitive to think of it as being mediated by a repressor. But once you see how the mechanism is, and I know you've seen this at least 3 or 4 times in your educational history, there is a repressor protein that binds to an operator downstream of the lac promoter, just like the arginine biosynthetic pathway that we just talked about. But the situation is different. Here the repressor protein, when it's not bound to an inducer, is in a conformation such that it does bind to the operator. So the lac operon, the lac I protein, the lac repressor, if lactose is not present, it exists in a conformation such that it binds very efficiently to that lac operator sequence. It binds to that double-stranded DNA just downstream and in fact somewhat on top of the lactose gene promoter, and it prevents, not completely, but it severely impedes the transcription of the lac operon, these genes lac Z, Y, and A downstream. Now, when that investigator in that previous graph dropped in that slug of lactose, that lactose gets into the cells and is actually converted. We'll talk about this again. Lactose by itself, the actual disaccharide, is not the inducer. There is an enzymatic activity that has to occur to change lactose into an isomer called allolactose. A little bit of a trivial fact. But this allolactose will now act as the inducer. So as allolactose binds to this repressor, it changes its conformation in just the opposite way that the arginine repressor did. The arginine binding to the repressor made the conformation such that the arginine repressor was now active. Here, in this particular case, the allolactose binding to the lac repressor makes it inactive. It falls off. And this makes perfect sense because now, in the presence of lactose, this delicious milk sugar that the bacterium has found itself in a bonanza of, it's now going to transcribe like a freight train all of the genes that encode the beta-galactosidase, which break it down. The LAC-Y gene, that's the permease gene. We used this as an example when we talked about symporters in the last section. LAC-Y is that membrane protein that uses proton motive force to simultaneously import lactose. And transacetylase, I still don't know if people know exactly what the transacetylase does, LAC-A. I think people have an idea. I'm not one of those people. But all of those genes will be transcribed only when lactose is present. So it has to do, again, with this negative control. So anytime you see a repressor, this is an example of negative control. And that makes sense because repressors repress things, so that's a negative thing. When we come back on Friday, we're going to talk about positive control. We're going to talk about activator proteins because activator proteins actually still work in the lactose operon as well. But we'll get to that on Friday, right? Friday.